Okay, now moving towards the completion of replication, as we have studied the initiation and elongation which is done with the help of primers and polymerases. We are moving towards the end of replication, that is known as the termination of replication. So before going to study the termination step, which is done with the help of telomerases, we will be encountering some other problems during the unwinding of the DNA, that is the formation of supercoils. What are these supercoils? The supercoils are actually the coils over coils. So when the DNA helicases move forward, it causes the DNA to wrap around itself. This problem is relieved with the help of some special type of enzymes called as DNA Topo Isomerases, which relieve the supercoils by Topo Isomerases. So if there are two types of Topo Isomerases there, because we will encountering different types of supercoils. If the supercoils are positive supercoils, then they are going to be removed with the help of topoisomerases 1, and if they are going to coil the DNA around the anti-clockwise position and causes the formation of negative supercoils, then we will take help, or the nature has designed another type of topoisomerases called as topoisomerases type 2 or DNA gyrases. So what is the difference between these enzymes? Topoisomerases type 1 acts by making a transient single cut into the backbone of the DNA, enabling the strands to swivel around each other to remove the building up of twists. Just like in this case, here you can see only one type of supercoils are there, so which occurs during the strand separation, and we encounter positive supercoils. This problem is solved by the help of topoisomerases type 1. What they're gonna do, they are going to make a nick or a cut in one part of the DNA, so the other part of the DNA will move from this side, and we will getting the straight DNA again. So we will be no more facing the problem of supercoiling. And what happened in the case of if the DNA having both positive and negative supercoils? Here in the second diagram you can see both positive and negative supercoils are there. So there comes the mechanism of action or the function of topoisomerases type 2. They act by introducing double-stranded breaks, enabling one double-stranded DNA to pass through another. So if we will make cuts at both ends of the DNA, at both sides of the DNA, on both strands of the DNA, they will be able to pass through another, thereby removing knots and entanglements that can form within and between the DNA molecule. Here you can see in this second diagram where both types of positive and negative supercoils are there, and the topoisomerase type 2 is going to make two cuts in both strands of the DNA. The left half of the circle folds over the right half. The back half of the helix is cleaved. The front half of the helix passes through the break, which is then released, and we will be getting the relaxed form of the DNA. So the mechanism of replication moves smoothly. Afterwards, the whole process, the whole machinery or the components of replication are now free. So nature is going to remove the primer and nicks are going to be sealed with the help of DNA ligases. So after the function of Topo Isomerases, we will be having another enzyme called as DNA ligase, which is going to cause the joining of the gaps and filling the gaps. Primers are removed by polymerase 1 by replacing ribonucleotides with deoxyribonucleotides, and nicks are sealed by DNA ligases. Multiple primers on the lagging strand while single primer on the leading strand. As I have told all this discussion in the previous lecture, that we have many enzymes or components during the overall process of replication. So here the function of all the machinery has done, and again the reconstitution of the chromatin structure occurs. But what is the meaning of reconstitution? Because during the process of replication, we causes the unwinding of the DNA. So after we got the copy of the DNA, we make the chromatin structure to reconstitute itself to gain the original position. So the chromatin structure must be reformed after replication. Newly replicated DNA is rapidly assembled into nucleosomes, and the pre-existing and newly assembled histone octamers are randomly distributed to each arm of the replication fork. As you know that DNA is coiled around the histone octamers and leads to the formation of first nucleosome and then to the chromatin structure. So the chromatin structure is reconstituted again. Here DNA synthesis and the cell cycle explains that which phase is responsible for which replication activity. So in animal cells, including human cells, the replication of the DNA genome occurs during the S phase. This is actually temporarily separated from the mitotic phase by non-synthetic periods referred to as the G1 and G2, occurring before and after the S phase. The cell prepares for DNA synthesis in G1 and for mitosis in G2. So here comes the last steps, which is the termination, which is done with the help of Telomerases, and the ends which are responsible for the termination of the replication are called as the telomeres. In eukaryotic replication, following removal of the RNA primer from the 5' end of the lagging strand, a gap is left. So this gap exposes the DNA strand to attack by the 5' end of the exonucleases. This problem is solved or overcome by the help of telomerases. So what he want to tell you is that during the formation of the lagging strand, the newly formed DNA strand becomes shorter in length as compared to the leading strand. So here comes the problem of shortening of the DNA, which is solved with the help of telomerases. This is the diagram in which the function and overall mechanism of action of telomerases is well explained. Here you can see the newly synthesized DNA where the lagging strand is full in length while the lagging strand is lagging some of the parts of the DNA, the brown one in color. So to overcome these problems, we have telomerases. The function of telomerases is just like the transcriptases, reverse transcriptases. As you know, transcription is the formation of RNA from the DNA. So reverse transcription is the formation of DNA from the RNA. So telomerases have the function of reverse transcriptases. What they're going to first recognize the telomeric ends of the DNA, and by forming the DNA from the RNA, it functions. Telomeres extends the 3' end of the DNA, and the RNA primarily synthesized by primase. Because at the telomeric end we need to start the DNA strand, so we need first primer onto which the telomeric ends are going to synthesize the further machinery of the DNA. What happens next? The 3' end of the RNA serve as an acceptor of the RNA polymerase. After the RNA going to initiate the synthesis of ribonucleoprotein or the RNA template, the polymerization is done again with the help of polymerases, which causes the formation of DNA from the single stranded RNA, which is known as the telomerase. So this is how the shortening of the lagging strand of the DNA is overcome with the help of telomerases, and afterwards at the end the RNA primer is removed, and we will be having full length of both the lagging and the leading strands of the DNA. This is the overall mechanism of action of telomerases, and telomeres may be viewed as mitotic clocks in their length in most cells, which is inversely related to the number of times the cells have divided. So the telomers or telomerases study gain very much important, especially in the studies of aging and cancer, because if the telomerases will not function properly, we will be having a short of many proteins which are not going to make, and the process of aging speeds up. And as these are the ends of the DNA, so anti-cancer agents are easy to gain importance at these sites. So the scientists are studying these areas to study or to find any clue about the treatment of cancer. So this is all about the replication process.